This project is called PrairieDNA, and it's about monitoring environmental DNA in Manitoba and Ontario. And the goal is to identify microbes and other species that are present in the environment that might be of interest to Indigenous communities. So we're working with First Nation communities in Ontario and Manitoba. So we're co-developing this project with First Nations communities in order to identify their interests and their community concerns.
Water is absolutely integral for sustaining any life as we know it currently. One of the very heavy foci of this project is on water and the health of water or aquatic ecosystems.
We know that communities that don't have access to safe water often tend to be at higher risk of getting a variety of infectious diseases. So how can we mitigate that problem would really help in improving the health of the community members.
My involvement is to look at antimicrobial resistant genes that could be present in water and wastewater treatment facilities in Indigenous communities, to evaluate the discharges from wastewater facilities and oxidation lagoons in these communities, as well as evaluate the water cycle in general from a microbiological perspective.
PrairieDNA is a pun because the title of the project is related to the Prairie's North region, which we've defined as the Lake Winnipeg Basin up to Hudson Bay. And eDNA means environmental DNA, which is DNA that we can find out in the environment. So that's a broad category that includes lots of different components of the ecosystem, but we're using it in this case to measure invasive species, harmful algal blooms, the presence of pathogens, and the natural microbial communities that are always there, but not very well known in the environment. The project has three themes. The first theme is to explore the microbial community diversity in natural waterways.
My own research is very much tied to the research theme 1 to this project, focused on the biodiversity exploration and understanding what the different microbial communities look like and how they change over time. Where we're basically trying to uncover any microbial novelty and at the same time trying to see how those microbial communities differ over time and trying to figure out if we actually need active conservation efforts for the biodiversity captured in those environments.
Since I came to Manitoba in 2019, I've been more interested in the freshwater systems and working with Indigenous communities in order to understand the changes that are happening, for example, with harmful algal blooms that are being observed in Lake Winnipeg and other community concerns about the health of the waterways here. Theme 2 is about monitoring invasive species in rehabilitated lakes. So this is a work with the experimental lakes area, and the goal is to rehabilitate lakes that have been overrun by cattails, replacing them with manumin, wild rice, and observing the changes in the health of the lake ecosystem after that rehabilitation. And then theme 3 is looking at the role of antimicrobial resistance genes and other potential pathogens in municipal water systems in First Nations communities.
My research is on looking at mechanisms of antibiotic resistance in bacteria. We are interested in antibiotic resistance because it is considered a silent pandemic. It kills about 1.3 million people every year. So looking at the prevalence of antimicrobial resistant bacteria in the environment is very, very critical when it comes to how to address the problem. And one way of doing that is by looking at the DNA that's present in the environment that may encode for various antibiotic resistant. The aspects that we are involved in this project will certainly help coming up with ways where we can improve the safety of the water.
My involvement in Team 3 deals with antimicrobial resistant and SKP pathogens. So to screen for antimicrobial resistance, we use molecular biology tools, and we can also use culture-dependent approaches in these aquatic ecosystems and water infrastructures. Our idea is to go from a plate that you will normally use to look for coliforms and E. coli and then use new tools available like nanopore technology, where you will load DNA into this tiny device, and then you will plug it into a computer, and then you will get DNA sequences, and then we will compare those DNA sequences against a database, like same way that you will do it for identifying a person.
This project will be co-developed with First Nations communities, so the intention is to monitor waterways that are in their jurisdictions. And so we have several community partners around the Prairie's north region, and those partners will be leaders in helping us guide the research. So it'll be particularly impactful for them because it's designed that way.
As a very strong focus in this larger project is associated with microbial community monitoring, or just community monitoring as a whole, that is nested within the human communities inhabiting these areas where these aquatic ecosystems are found.
We're looking for SKP, which are bacterial pathogens that can be connected with clinical cases occurring in these communities. To assess for these pathogens in aquatic ecosystems of these communities, we need to use these plates to isolate these colonies, because they are live isolates that we're gonna aiming for. And then we will go from these colonies to know the full genome using this device, right? That's another component of team number 3, that we are doing this in collaboration with two First Nation communities.
What happened is that we applied for one proposal, then it got rejected, but a second one in this stream came up, and we were able to take advantage of having already worked together on the previous proposal to get this one in quickly. So it was serendipitous, I would say, that there was an opportunity that came along at the right time. And having already known the other members of the group from other projects and through the university system, it was good that it all came together on such short notice.
I think one of the cool things about being involved in this project is the opportunity to work with people with different expertise, and I do believe that this is how science should be done. Science is done best when it's done together with people who bring in complementary expertise. So I'm really looking forward to being part of this team, and I am also particularly looking forward to interacting with young scientists, graduate students, undergraduate students who will be part of this project.
There's funding for graduate students in the grant, so there will be opportunities through the various principal investigators that are involved in the project.